Today we'll be discussing the breakdown of proteins becoming amino acids and their paths undergoing the process of either oxidation deamination or transamination occurring in the liver. So first, proteins will be eaten, which then they travel to the stomach. Here they are mechanically broken down with the help of HCl and pepsin as it makes its way into the duodenum. Here, the S and the I cells are activated and they travel through the bloodstream, which is here, and then they travel to the pancreas, which then releases a signal to release zymogens back into the duodenum and also bicarb as well. This then activates trypsin, which then turns on chymotrypsin, carboxypeptidase, and aminopeptidase. Those are the enzymes used in this process. This then hydrolyzes the protein into dipeptidase, tripeptidase, and amino acids. Then it travels into the jejunum here, and then at the brush border of the jejunum, you have the assisting enzymes, aminopeptidase, carboxypeptidase, and dipeptidase. These enzymes help the amino acids enter the enterocyte, which is here on the brush border of the jejunum, which then exits. They also help the amino acids exit the enterocyte into the bloodstream, and then they travel into the liver. So first, let's talk about the essential amino acids needed for your body. So there are 10 essential amino acids that your body needs, but unfortunately, the body cannot make these essential amino acids, so you need to consume them through food as they're important building blocks for your tissues. So once the amino acids reach the liver, there are two processes that occur, either transamination or oxidative deamination. So in a healthy individual, they will experience transamination 85 to 90% of the time. So this is when you're recycling the ingested protein by transferring an amine group. So you will transfer NH2 and in exchange for O2 from the keto acid group to form new amino acids, which then go into the nitrogen pool, which are recycled and turned into ATP. This process is facilitated by the transaminase enzyme, and so that's the E here, so that's transamination. So then the healthy individual will also go through oxidative deamination 10 to 15% of the time. So this process is when the liver is broken down, when the amino acid is broken down in the liver, and there's an excess amount of protein intake. So when there's an excess amount of protein over here, and it can't go into the nitrogen pool, it is then over to oxidative deamination. So the amine group is removed from the amino acid and converted into ammonia, which is here NH3. So then glutamate is a nitrogen carrier, which then takes the NH3 ammonia and picks up a hydrogen ion, which then converts it into ammonium, NH4. So then NH4 goes into the mitochondria, which then, with the help of ATP, the NH4 then is converted and goes into the urea cycle, which then the urea goes into the bloodstream, which then travels to your kidney, which then is converted into urine, which then is excreted out of the body. So now let's recognize what may occur when the individual is unhealthy. So this is it when you're experiencing, more deamination than transamination. So the NH4 in the liver, after ammonia and glutamate pick up a hydrogen ion, NH4, this is when the NH4 is not able to enter the mitochondria like normal due to incorrect signaling the body is receiving. So this then causes the NH4 to go into the bloodstream, which then reaches the brain. So the glutamine enzyme then breaks down the ammonium and the glutamate into glutamine and inhibits the GABA neurotransmitter along the way as well. So then this damages the mitochondria and produces brain degradation. So when an individual continually undergoes this process of deamination instead of transamination, they are not using the protein correctly to fuel the body, which will then cause them to have a decrease in energy and then just many, many other things going wrong.